Okay, now we're recording. Can everybody see the slides okay? Okay. Let's go ahead and pick up. So last time we were talking about the pigments, and we were talking about a little bit about the fall colors. And so now what we want to do is we want to start zooming in on what this all means for us. So remember what we're talking about is the pigments absorbing light. And so we want more than just one pigment doing that. So this is kind of where we get what's called this antenna complex, where you have lots of different types of accessory pigments, such as chlorophyll, and their job is all doing the same thing, basically gather light in different areas of the visible spectrum. And then you'll be able to capture the energy from that light, and it'll basically funnel it into the reaction center. So a lot of times the antenna complex works like a funnel. An energetic funnel. So essentially what happens is you kind of have this sort of area where you're basically absorbing light, and it basically kind of grabs onto that light, absorbs that light. The pigments absorb the light, and then they basically pass the energy associated with that light from one pigment molecule to the next, to the next, to the next, until eventually what happens is you get down to your reaction center, which is your chlorophyll A. And so that's basically where all your energy is going, is into that chlorophyll A. So other than that, you're basically kind of capturing all this light from this really broad and huge complex, and it's basically sort of transferring all this energy and sort of funneling it, if you will, into the reaction center, which is where you have your chlorophyll A molecule. So your chlorophyll A molecule is probably one of your most important pigments because that is your reactive core. That's where you're basically going to get your excited electrons. So everything is going to this chlorophyll A molecule, and in particular into the porphyrin ring associated with this chlorophyll A molecule. And so the energy then goes into the actual electron, and this excited electron then enters into the photosystem where it gets captured, and then you basically sort of start transforming that energy step by step into different areas. And so that's kind of what's happening in this photosystem. So when you take a look at your antenna complex, this is basically a giant construction or a giant complex of pigments that's designed to harvest light. And so it's going to be capturing all the sunlight energy and basically funneling it into this reactive chlorophyll. And so basically it really does what it says it does. It basically harvests light. And then ultimately it's going to funnel it down into this chlorophyll, this reactive chlorophyll. And this is all found, by the way, in the membranes of the thylakoid. That's basically where everything is found. So here's kind of what a photosystem looks like. So this is essentially your big blob of pigment. So this whole thing here is essentially your antenna complex. It's your complex of pigments, all sorts of different types. So you basically get a photon of light that comes in. It gets absorbed by these pigments, and then the energy associated with that will be absorbed in the electrons, and then the energy from these electrons are basically passed on from one pigment to another, to another, to another. It's basically bouncing around through this entire antenna complex until eventually it finds its way into this reaction center. So this is your chlorophyll A. And then when it excites that electron, then it jumps out of that chlorophyll A. And then you have another protein typically, which is an electron acceptor, which will snatch this excited electron and basically send it off into the biochemical pathways of the photosystem. And so basically what happened is, in this case, is once this occurs, this basically experiences a net loss of the electron. Because that excited electron gets snatched up by this electron acceptor, and it gets moved on into the photosystem. Now, the problem with that is, since you've lost an electron, what this basically means is this now exists in an oxidized state. In particular, actually, your reactive core exists in an oxidized state, which is a very unhappy place to be. And actually represents a bit of a problem, right? Because if you think about it, nothing has an infinite supply of electrons, because if you just keep coming in and keep beaming this thing with photons of light and it keeps exciting an electron, eventually you're going to run out of electrons because things only have a finite number of electrons. So when you lose this electron, then you have to replace it. And that's where you get your electron donor from, is the electron donor will then replace that electron with another electron so that you can excite it again, and you'll lose that excited electron, and then you'll have the donor backfill that extra electron. So as long as you have a donor who's willing to supply an electron, then you can keep the system going. The second you lose that electron donor, then you're going to basically run out of juice. You're not going to have an infinite number of electrons to move through the system, and so you're going to basically be dead in the water. So this is how your basic photosystem works. Now let's take a look at this reaction center, right? Because we said that this reaction center, which is a chlorophyll A reaction center, will absorb your photon of energy. It will basically create the excited electron, and that'll jump up into a higher level. And then that electron is going to be transferred to your electron acceptor. So basically it leaves the chlorophyll. And once it leaves the chlorophyll, then you have an oxidized chlorophyll, which is very, so it's got like a hole. It's got a gaping absence there associated with the loss of this electron. And what it wants to do is it wants to fill that hole. So basically what this makes this a very aggressive oxidizer, because it wants to fill this hole. And so it'll basically take an electron from anybody. So it's actually one of the strongest oxidizers that we know of, even stronger than some of the oxidizers that we generate from oxygen consumption in cellular respiration. This is a very strong oxidizer and a very nasty molecule. You don't want to be on the business end of this one because it is not happy. It's going to basically take your electron from you and just basically leave you behind to die. That's pretty much what's going to happen. So this is just kind of a schematic of sort of like how it basically plays out. So essentially what you have here in blue is your electron donor, in orange is your electron acceptor, and then of course you have your photosystem in green. So typically what happens is light will come and it'll hit your green photo system, and then it'll hit that electron, excites the electron. So here's your excited electron. And this excited electron, then what's going to happen here is this electron is going to go to the electron acceptor. So the acceptor becomes reduced and the chlorophyll becomes oxidized. So it has kind of like this really strong oxidizing presence here. That's a positive charge. So in order to satisfy this oxidized chlorophyll, what happens is your electron donor, which also has an electron to give, will basically give that electron to your chlorophyll, and then the chlorophyll will be happy again because now you've supplied it with an electron. And then basically that resets your system back to the beginning, where you can essentially excite or beam energy into another electron in your chlorophyll molecule. So you can see that basically it's just kind of a cycle. This just sort of keeps going, as long as you have a steady source of electrons coming from your electron donor. So when you take a look at your light reactions, right, your basic protocol that you look at is essentially your photon of light, which is going to be absorbed and captured by a pigment molecule, where it's going to excite that electron. So the energy is going to go into that reaction center eventually. It's going to excite that electron, which is going to be snatched away by this acceptor molecule. So this basically takes the electron away. As a matter of fact, in this thing here, notice we still have the electron here, but in reality, this electron goes away and gets passed on, so it's being used in other pathways. So it doesn't, hang around to just go right back to the chlorophyll. It gets taken off into this electron transport chain. So basically the electrons associated with this excited electron then is going to go through a type of transport chain. So this is an electron transport chain similar to what we saw in cellular respiration. And this is going to be what drives this electron transport chain, is going to be what drives the energetics associated with, in this case, the production of NADP+. So some of those electrons will actually dump onto NADP+, right? So that's one piece of a similar story that we've seen in cellular respiration. In this case, it's kind of a photosynthetic version of it. So this NADP+, remember, this is also another one of those electron taxis where you basically have room for, so NADP+ for electrons. And then when you add those electrons to it, then what's going to happen is you're going to create this reduced form, which is NADPH. And so here you can have your electrons bound in this situation. And then, of course, it's going to go the opposite direction as well, just like NAD plus and NADH. It's the same exact situation that we have here. It's just a different cab company, that's all. So you basically have your oxidized form and your reduced form. And so part of the electrons are going into that. But then it's also setting up a concentration gradient, which will allow you to produce ATP by way of a proton gradient. Similar to what we saw in the mitochondria in cellular respiration. So you're doing the exact same thing. Only in this case, it's in photosynthesis. Now, there is a sort of an offshoot of photosynthesis, what we refer to as cyclic photophosphorylation. So this typically happens in sulfur bacteria. Um, and so this is a situation where you don't utilize oxygen in photosynthesis. So this is kind of like that older version of photosynthesis where you're reducing sulfur instead of oxygen. And so here you basically have a similar thing, because remember, sulfur and oxygen are in the same family, right? They're in the group 6 family, so they have very similar chemical properties. So in this case, this is sulfur acting very similar to oxygen, only you don't have oxygen, so you have sulfur, and it's setting up the same thing. You're setting up the same electron transport chain. You're using the same proton gradient to drive ATP synthesis, only in this case, it's a sulfur-based system. And this is kind of what it looks like. So here you can see a photosystem which excites your electron. All that's the same. You basically snatch that away with your electron acceptor, so that's all the same. In this case, you power a BC1 complex, which is a proton pump. That's basically what this is. And instead of being linear, so what we're going to focus most of our time is linear, but we're just going to basically kind of clean up the cyclic thing. So instead, at this point, this is kind of where it's different. So in this case, in cyclic, what happens is the electrons get dumped onto an electron acceptor, which then will dump those electrons back into the reaction core. So what you essentially do is you take the electron, you kind of wring out all the energy that you absorbed in that electron, and you return that electron back to the reactor core. That's kind of what cyclic photosynthesis does. We don't really see this a lot. So most of what we actually have on the planet, plants and so forth, our major photosynthesizers, are typically associated more with oxygenic photosynthesis, not sulfur photogenesis. So this is cyclic photosynthesis. So what we want to focus most of our time on is oxygenic photosynthesis. That's basically when we have oxygen molecules around, because that defines the majority of our photosynthesizers and our producers in the world. And so we have, in this case, we've already talked about the construction of the photosystem. We have now the two different photosystems actually in place. So when we're talking about sort of traditional types of photosynthesis, we have essentially two photosystems in series. One is what's called Photosystem I, which is very similar in its functionality to sulfur bacteria. The only difference is it uses oxygen as a source of its electrons instead. This one basically we refer to as a P700, so Photosystem 700. This is basically referring to the wavelength of the pigment that's in there, of what it absorbs, basically, the wavelength of light that it absorbs. And then we have a second photosystem, Photosystem II. This is the oxidizer, right? So Photosystem II is oftentimes referred to as the water splitting photosystem. And this one we refer to as P680 because that's the wavelength of light that it absorbs. So when you put the two of these together, Photosystems I and Photosystems II, then the two are a very powerful combination because Photosystem II will basically split and oxidize water, take the electrons from there, and then basically funnel the electrons through the rest of the system and feed those electrons to Photosystem I. So essentially what you get is a sort of linear, non-cyclic transfer of electrons, very similar to the way you see cellular respiration in mitochondria. And so this is sufficient to generate both ATP and NADPH, both using the electron transport chain and chemiosmosis. So when you take a look at Photosystem I, The goal of photosystem 1 is to make NADPH. So what it does is basically it transfers the electrons ultimately to NADP plus into NADPH. And so there you will basically satisfy one of the two requirements for the light reactions. Remember, the light reactions are taking oxygen and sunlight, or water rather, water and sunlight, and it's basically turning that into ATP, NADPH, and oxygen. That's its byproduct. So this is one. So photosystem 1 is responsible for creating NADPH. And photosystem 2, basically what you have is the complex that will make ATP. So this is what it's tasked to do, is photosystem 2 makes ATP. It also is the source of electrons. So since photosystem 2 is splitting the water and creating electrons from the water, then it passes these electrons on to photosystem 1. Because remember, photosystem 1, you're also losing electrons. So the question is, how do you backfill your electrons in your reactive core in photosystem 1? Well, the way you do that is with photosystem 2. So photosystem 1 gets its electrons from photosystem 2. However, the question then is, where does photosystem 2 get its electrons from? It gets its electrons from water. So water is actually the source of electrons that feeds photosystem 2 and replaces the electrons that basically exit photosystem 2 when it gets excited and it absorbs that energy. So when you take a look at this, then the two halves, we have photosystem 1, photosystem 2, and their transport chains are connected by a proton pump called B6F. So cytochrome B6F complex, which is a proton pump, which of course, you know, all proton pumps do the same thing. They basically make proton gradients, which are energetic scenarios that helps us to basically fuel the energetic needs of ATP. That's what they do for a living. So most of our photosynthesis is going to use photosystems 2 and 1 in a series, in which case it's a linear pathway. And so we kind of write this out. It starts off with the splitting of water, right? That's the source of electrons. And so we basically then create with linear photosynthesis. We refer to what's referred to as the Z diagram. So this is the Z diagram. So you can see there's a lot going on here, but you can see it looks kind of like a Z on its side. So basically that's why I call it Z diagram. So the first thing you're going to notice is we've got two photosystems, photosystem 2 and photosystem 1. We tend to think of this as sort of starting off with photosystem 2, not because, and the reason why this is kind of backwards is because we discovered photosystem 1 first and photosystem 2 after photosystem 1. So they kind of kept their numbering conventions, even though technically speaking, in the series, photosystem 2 works first instead of photosystem 1. The important thing to understand is when you have these photosystems embedded in the thylakoid membranes, they're both absorbing light energy. So they're both absorbing photons of light. So at the same time, they're both getting light, which means they're exciting the electrons in the pigment molecules, which then transfers the energy from one to the other, to the other, to the other. Same thing's happening in both in photosystem 1 and photosystem 2. That energy then goes into the reaction center where it's absorbed by the electrons, and the excited electron then jumps up into a higher order state. Not physically, but electronically or chemically, it jumps up into a higher energy state. Same thing's happening in photosystem 1, where the electron gets excited and that electron jumps up into a high energy state. Then, in order to kick off the electron transport chain that we see here, we have a reaction center, or the electron associated with the reaction center, being picked up by an electron acceptor. In this particular case, the electrons are picked up by a molecule called plastoquinone. And so that basically grabs a hold of the excited electron that comes off of the photosystem 2, and then plastoquinone will take that electron and transfer it to the B6F complex. The B6F complex then, just like the proton pumps we learned about in cellular respiration, will take a cut of the energy associated with those excited electrons and utilize that energy to build a concentration or a proton gradient. This will be then used by chemiosmosis to fuel the production of ATP. So you can see the exact same mechanism of electron transport chain plus chemiosmosis is at play here in this first bit, or this first half of the photosynthetic activity associated with photosynthesis. Now, in order to backfill the electron that this photosystem II reaction center lost, basically this reaction center will split a water molecule, turn it into hydrogen and oxygen, rip the electrons off, and then it'll basically fill those electrons into the reactive core of photosystem II, thereby replenishing your source of electrons so that you can basically excite those with another photon of light. This, by the way, is one of the reasons why you have to water your plants. You water your plants because you have to have a source of electrons in order to fill in these reactive cores. Otherwise, they're just not going to be able to do any photosynthesis. Now let's go ahead and loop back around to the photosystem I. Remember, the photosystem I's electron also is excited. It then gets snatched up not by plastoquinone, but by a molecule called ferredoxin, which will grab onto that excited electron. And then ferredoxin will pass those electrons on to NADP reductase, which will then fill an NADP plus with the electrons, and then that will basically convert it into NADPH, remember, which is the reduced form of this particular molecule. But the reaction center for photosystem I is now empty. It has to be replenished. How does it replenish it? Well, plastocyanin, which has inherited the electrons from the B6F complex, will then dump those electrons into the reaction center in photosystem I, therefore replenishing its supply of electrons. So where those electrons come from? They were the original electrons that came from the reaction center associated with photosystem II. So photosystem II will actually feed photosystem I and replenish its electrons. The electrons of photosystem I end up in NADPH, but ultimately what kicks it all off, the ultimate source of electrons, is water. So the electrons in water get taken into photosystem II's reaction center, which gets excited, it jumps up, goes through the electron transport chain, and then basically is used to backfill the photosystem I loss of electrons when plastocyanin gives that electron to the reaction center for photosystem I. Then they get excited and they jump up, and then those electrons get delivered to NADP plus to become NADPH. So ultimately, the electrons trace their pathway from water to photosystem II, through the electron transport chain to photosystem I, then to ferredoxin, and then into NADPH. And then between the two of these, with your proton gradient, you will have made ATP, which is exactly what you wanted to do with these reactions, and also you will have made NADPH, which is also what you wanted to do with these reactions. So in addition to this, your byproduct is oxygen, which is the reason why photosynthesis gives off oxygen whenever you are, whenever the reaction is going forward. It's a byproduct essentially of photosynthesis. So here is basically a slide that looks at the effect of light on photosynthesis. So here you can see on the y-axis, you have the rate of photosynthesis versus the amount of time versus the light that you're actually turning on. So for instance, in one case, you have what's called far red light. That's kind of over toward the edge of the visible spectrum. That's infrared light. So when you turn the light on, notice you get a certain level of photosynthetic activity, and then you turn it off and the photosynthetic activity goes down. So you basically have this little bit of a bump up. When you're more solid in the red, you're not quite over the infrared side, you notice that you get the same thing, basically a little bump up of photosynthetic activity, and then it comes back down. Then when you put on both lights, far red and red, so basically more visible light is absorbed. You can see that you get a huge jump up in photosynthesis, suggesting that you're absorbing more light. And so you can see how the strength of having multiple pigments is actually quite strong, that it gives you a great deal more absorption of light than any of them by themselves. Now, then when we take a look at photosystem II is very similar. So remember we said photosystem I resembles sulfur bacteria. Photosystem II kind of has some resemblances to purple bacteria. And so what we have basically is 10 transmembrane proteins. That's kind of like that reactive core. So this is basically a description of the reactive core. So we have these 10 transmembrane protein subunits. And so they're going to be doing this electron transfer piece. And you have two P680 chlorophyll molecules. Remember, the P680 is associated with the wavelength of light that they absorb. So this is your chlorophyll A stuff. So in addition to this, the purple bacteria, and the reason why it's purple is because what it has actually in its core is manganese, which is actually a purple ion by itself. And so this is what allows us, this sort of high oxidative chlorophyll, is P680, is what allows us to be able to oxidize water. And then, of course, we have in Photosystem II our B6F complex, which is a protein pump. And this is all embedded in our thylakoid membranes, right? That's where all the stuff is. Everything is going to be embedded in our thylakoid membranes. So the Photosystem I then is basically going to have a complex of transmembrane proteins, a little bit more, between about a dozen-ish or so of these subunits. They're going to be bound to two P700 chlorophyll molecules, so slightly different in absorption spectrum. So they're going to be absorbing a slightly different wavelength of light. And so this is designed to basically take an electron from plasticine. This is the one that basically takes the electrons that were kind of going through the Photosystem II system and basically passes it on to and fills in the hole created by the loss of electron in Photosystem I. And then ultimately those electrons will find their way onto NADPH. So that's basically what happens. So this is, they always say a picture is worth 1,000 words. And so that's kind of what this is here for. So here you can see your two photosystems, right? You can see Photosystem II, and then you can see Photosystem I in this particular case. And so these are going to be absorbing light, and they're going to be absorbing it at the same time. And so they're going to be activating their reactive centers, and they're going to be passing those electrons on to plastoquinone, first of all, in Photosystem II, and then that's going to be passed on to your complex, your proton pump. So you can see it's pumping out a proton as it takes a cut of that energy in your electron. So that's your B6F complex. Then it passes it on to another shuttle protein, plastocyanin, which will then pass it into and fill in Photosystem I. So Photosystem I has lost its electron and passed it on to ferredoxin, which then feeds it to this NADPH reductase. And so that basically takes the electrons and loads it into NADPH, which is out here in the stroma. So that's where NADPH is being made. Now, the proton gradient, just like we saw with cellular respiration, is going to be transferring its energy into chemiosmosis. So that's what this ATP synthase is here. So here's your chemiosmosis. And so that is going to basically go through the ATP synthase, which is going to power the production of ATP. So now notice both your ATP and your NADPH are out here in the stroma, or the fluid-filled region of the chloroplast, whereas everything else is happening in the thylakoid membrane. So chemiosmosis then is driven by the ATP synthase enzyme, just like we saw with cellular respiration. So the protons are what basically drives this. So it basically moves through the ATP synthase, and it basically transfers its energy into the production of ATP. And so ultimately then, this means that ATP and NADPH are out in the stroma, and the stroma happens to be where the Calvin cycle reactions are, the dark reactions are. And this is essentially the carbon fixation piece, which requires ATP and NADPH in order to go forward. This is one of the reasons why you have everything organized so that your NADH and ATP are in the stroma, because that's where they're needed. And so once you actually have your NADPH and your ATP, then you can actually go to building your organic molecules, in this case, your carbohydrates, and you can essentially make your sugars. That's essentially what you're going to be doing. And NADPH and ATP are the energy sources for that. So you can kind of play around with this. So with cyclic phosphorylation, it's kind of like a short circuit of your normal linear pathway. So let's take a look at the Calvin cycle, so named from Mel Calvin, who had a SWAT team of people who worked on the Calvin cycle and figured it out. So the Calvin cycle is where you make your sugars. This is what you wanted to do, right? Everything else was preparation for this point. This is where you wanted to be. This is what you wanted to do. And so in this particular case, what's going to happen is you've generated ATP from light energy. So you've successfully captured light energy and you've sequestered it into ATP. So that's a good thing. This will then drive this endergonic reaction. And then you also have the reduction potential of NADPH from the photosystem I, and this will allow you to kind of use it as the same way you had with the electron transport chain as a source of energy, where you can dump these electrons and sort of create this sort of this kind of proton gradient. So Calvin cycle, named after Mel Calvin. Also, we refer to it as C3 synthesis because it uses a 3-carbon molecule. But this is kind of like standard, what we refer to as standard photosynthesis. So if you're kind of learning photosynthesis light on YouTube, oftentimes what you'll run into is this C3 or standard photosynthesis. And this is basically a situation where you take a carbon dioxide and what you want to do is basically build it into a carbohydrate. And the way you do that, similar to the Krebs cycle, right, or the citric acid cycle, is you need an acceptor molecule. In this case, our acceptor molecule is RUBP, ribulose bisphosphate. And so what's going to happen is you're going to take your ribulose bisphosphate, you're going to sequester a CO2 to it, a carbon to it, and that's going to form what's called PGA, phosphoglyceraldehyde. And the enzyme that does this fixation piece of it here is what's referred to as ribulose bisphosphate carboxylase oxygenase, or Rubisco is its name. Now notice it's important, the C is carboxylase and the O is oxygenase. So it's important to understand that this has two different personalities to it. Rubisco will basically bind to CO2, and also it will also bind to oxygen. So it does both. It's a bit of a switch hitter there. And so you obviously, one is helpful, the other one is not. So for instance, if you're doing photosynthesis, you don't want it to do this. You do want it to do this. You want it to bind to CO2. You don't want it to bind to oxygen, because CO2 is what you're trying to use to create your sugar with. Oxygen for you as a plant is a waste product. So you don't want to bind your waste product. So in Calvin cycle, when we take a look at Calvin cycle, there's 3 major phases of Calvin cycle that kind of help you organize exactly what's going on with the Calvin cycle. The first of those phases is what's referred to as carbon fixation. So this is a situation where you take your acceptor molecule, RUBP, ribulose bisphosphate, and you attach a CO2 to it to create phosphoglyceraldehyde. And who's doing this? Rubisco. Rubisco does this, the enzyme does this. And then that gives you basically carbon fixation. So you fixed your carbon into the cycle. That's basically why they call it carbon fixation. Then once you actually get it in PGA form, then you want to go through what's called reduction. So you reduce it from phosphoglyceraldehyde into glyceraldehyde 3-phosphate. So they sound similar, but they're different actually. So G3P is what we want. That's where we want to go. So it's a 3-carbon molecule at this point. And so with the next step then is regeneration. So you want to regenerate your acceptor molecule, similar to we saw the regeneration phase of the Krebs cycle, generating oxaloacetate or oxalate basically in order to have another acceptor molecule. We do the same thing here. Regeneration of RUBP requires 3 of these molecules basically in order to actually there's more than that, several of these molecules in order to generate an RUBP. So when we take a look at this, then we have these 3 basic systems. We have carbon fixation, and then reduction, and then the regeneration of RUBP, your acceptor molecule, so that you can go and fix another molecule of carbon dioxide. That's basically how you kind of keep the cycle going. So once you go through this 3 times, you can generate enough carbon to produce a new G3P. And 6 turns of this is enough to generate 1 glucose, which is what you want. This is right here what you're after, your glucose, which is your 6 carbon carbohydrate that we use for respiration, cellular respiration. So it's kind of what it looks like. So here you can see Rubisco is going to take your RUBP and it's going to attach your CO2 to it, and then it's basically going to turn it into 3-phosphoglycerate. And so you have several of these, so 12 molecules of these, for instance. Your 3-phosphoglycerate then is going to turn into 1,3-bisphosphoglycerate. Don't worry about the names too much, right? But you're going to consume some ATP in this process, right? So you're going to be, it's an energetic process. That's the ATP that we made. This is what was made by the light reactions. That's why you make ATP in those light reactions. And then this 1,3-bisphosphoglycerate is going to be converted then into your glyceraldehyde 3-phosphate. This is your G3P. That's what you want. But you're going to consume your NADPH. And that was also made in your light reactions. So notice you're making these two molecules in your light reactions because you need the energy in them in order to drive the Calvin cycle. So this whole part from going from PGA to G3P is the reduction phase. So you've been reducing it. Now you can take a couple of G3Ps and you can assemble them together to create a glucose molecule. However, we need to regenerate this, right? So we need to regenerate or rebuild RUBP because we can't be without our acceptor. Otherwise, photosynthesis is dead in the water. So what we do is we take 10 of these G3Ps, consume some more ATP, and then it's going to basically give us ribulose bisphosphate. So you can see the multiple arrows here. That means there's several steps in there that we're skipping for sake of clarity and brevity, basically, is the reason why we're doing it. And so that's basically what's happening with Calvin cycles. So essentially, it's very similar, has a lot of reminiscences to the Krebs cycle. In this case, you're fixing carbon, you're reducing it to G3P, which you then use as a carbohydrate, and then you regenerate the RUBP in order to be able to go through another round of carbon fixation. So when you take a look at it then, ultimately of Calvin cycle, glucose isn't the direct product of Calvin cycle, G3P is. And so you can shuttle G3P around in the plant cells. Matter of fact, when we said that the sugars are loaded into the phloem earlier in this chapter, what's actually loaded in the phloem is typically G3P because it's easier, it's more transportable type of, it's a disaccharide basically. And so it's a little easier sort of a thing to carry around and sort of transport. However, you can put two of them together in order to make a glucose, and you can, of course, you can stitch these glucose together to create your insoluble glucose polymer, which is essentially your starch, and then, of course, that's your storage for later use. So if you're making tons and tons and tons and tons of carbohydrate as a plant, and you're not needing it right about now because you've got good root growth, and so you got a ton of it, then you can start storing this G3P in the form of starch. So we take a look at the energy cycle. We kind of like to sort of fit it all together with what we do with the essentially what cellular respiration does. So photosynthesis, for instance, basically takes the products of respiration as a starting material and vice versa. So for instance, when you take a look at mitochondria, we take the byproduct of photosynthesis as our starting point. We need oxygen in order to do cellular respiration, which is the byproduct of Photosystem II, which is the water splitting photosystem. One of our byproducts is water, right? Because oxygen is our terminal electron acceptor. So we generate water vapor, which is something that Photosystem II needs as a source of electrons. So you can see our outputs, our trash is their treasure, and their trash is our treasure. So the two systems, actually, photosynthesis and cellular respiration, really do fit together very nicely. They lace together very nicely. And then, of course, if we generate CO2 in Calvin cycle, you need CO2 in order to be able to fix it to create glucose itself. And of course, we need glucose in order to generate our energy because we don't make our own energy. So we need that glucose. So you can see how photosynthesis and cellular respiration, the activities of the chloroplast and the activities of the mitochondria go very much hand in hand, and they complement each other. So it's very much kind of like lacing your fingers together, that your two hands, right and left, fit together perfectly. They lace together very nicely. And the same thing is also true with chloroplasts and mitochondria. Now let's talk a little bit about a bit of a problem that we have in terms of photosynthesis. So we talked a little bit about C3 photosynthesis, which is kind of standard photosynthesis. Now we want to talk about one of the weaknesses or the problems associated with the enzyme that we have in Calvin's cycle, which is Rubisco. So remember we said that Rubisco had two different functionalities to it. It had carboxylation, right? The carboxylase, oxygenase. It also has this oxygenation piece of it. And so where this is what we want Rubisco to do, this is often what it will do if we put it under undesirable circumstances. So for instance, if we have a lot of CO2, then RUBP will take that CO2 and it'll convert that, it'll fix that CO2 into the carbohydrate, and we'll have plenty of carbohydrate um, to work from. So that's a good thing. This is what we want, right? However, because Rubisco has two functionalities to it, it's also got oxygenase. It also binds to Rubisco, oxygen does. And so when it binds to oxygen, it's what we refer to as photorespiration. It's the oxidation of RUBP, or the addition of oxygen. This basically is not useless. This is not beneficial. This doesn't help photosynthesis. So if your Rubisco is binding a lot of oxygen but not CO2, then you're not doing photosynthesis. So you're not eating. So this is obviously a problem. Well, how's this a problem? Well, it's a problem because in some situations, we may be in environments where we have a low CO2 and a high oxygen level. In which case, if you have a high oxygen level, then your Rubisco is going to bind preferentially to oxygen, and it's not going to bind to CO2 because CO2 and oxygen are competing for the same site on Rubisco. Think of it this way. Think of it as two people fighting relationally for the same person, like for the same man, right? It's two men fighting for the same woman, or two women fighting for the same men, right? So one thing that Rubisco is basically a little bit of a switch hitter. So Rubisco has a bit of a rangy eye. So this is a bit promiscuous. And we actually do use that word, promiscuous, to describe enzymes that don't have a lot of fidelity to their substrate, right? And fidelity conjures up the ideas of infidelity in marriage and things like that, right? And so the idea here is, it's kind of like let's say if you're a girlfriend and you've got a boyfriend with kind of a bit of a wandering eye, and he doesn't have any problem cheating on you, then one of the ways you can keep your boyfriend from cheating on you is by eliminating his access to other women. And so in that case, he's faithful to you, right? Because you're kind of like the only person that he's around. However, that doesn't necessarily mean he's faithful to you. It just means that you're the only person he's got exposure to. Why? Because if you go in, take him into a big room where you've got all sorts of other women, then all of a sudden you find that, he's off traipsing around with all these other women, and you're kind of sit here like, okay, wait a minute, but it's like I'm supposed to be the one that we need to make a connection with, and you're sort of traipsing around with all these other women. Why? Because you've kind of got this promiscuous eye. Rubisco is very promiscuous that way. So the only way that you can actually keep Rubisco faithful to photosynthesis is if you basically eliminate its exposure to oxygen. And so if it's got oxygen around, then it starts to traipse around with oxygen. And the more oxygen you have, then the more it traipse around with it. So like, for instance, if it's just you and one other woman with the man, then, I guess theoretically speaking, you could probably say you got a 50-50 chance. If you've got, you and say 100 other women, then the chances are that you're probably going to lose out more often than not. Why? Because there's more of them than there are of you. And that's very much the way Rubisco is, and how most of these are. Why? Because they're a competitive interaction, right? They're competing for the same active site. This is a type of competitive inhibition. So how do you deal with this? And here's the problem with this, right? This is the reason why this is a problem. Because this is a problem when you have hot conditions. So let's take a look at normal oxygen CO2 exchange. So oxygen CO2 exchange happens in a leaf through the stomata. This is basically like the lungs of the leaf. Now, normally with stomata, CO2 is supposed to go in and oxygen is supposed to come out. However, here's the problem. Because you've got a lot of water in you and you're taking up a lot of water and you're a moist environment, there's also water inside of you. So what that basically means is if you have hot, arid conditions on the outside, then water is going to come out into these hot, arid conditions, right? By osmosis, essentially. And they're going to want to come out. So that's a bad thing, right? Because if the water comes out of you as a plant, then what's going to happen is you're going to dehydrate and you're going to die, right? So you don't have that much water because usually if it's hot and arid, there's not a lot of water for you to absorb in your roots. So you don't want to certainly lose water. So there's not a lot of water in the soil. So this is like in a C3 situation. So in a C3 situation, what you do is in order to preserve water, then you have to close your stomata. And so the problem with that is closing off your stomata. So here's your C3 situation. So basically what's going to happen is when you close your stomata, that's going to basically cut off your CO2 supply. So you don't have CO2 to do photosynthesis with. However, you do cut off the flow of water, so you reduce water loss. That's good. But you also increase your oxygen inside. That's not good. Why? Because the more oxygen you have inside, the more likely your rubisco is going to bind to the oxygen, not to the CO2. If it binds to the oxygen, not to CO2, then it's not going to be doing photosynthesis. And so you're going to basically stop. Your photosynthesis come to a grinding halt. So this is basically when your stomata are closed. Why would you do this? Because if your stomata are open at this point, here's what's going to happen. Yeah, you'll get a lot of CO2 in. That's true. And you'll reduce your oxygen. Yes, that's true. Those are all good. That's good. That's good. But you're also going to really lose a lot of water. That is bad. That's a double, triple, quadruple bad. Because losing your water is basically losing your source of electrons, and that'll also kill your photosynthesis. Do you see the nature of the catch-22? It's kind of like a damned if you do, damned if you don't sort of a thing. If you close your stomata, then you're going to be retaining your oxygen, which basically reduces your ability to do photosynthesis because you're not getting CO2. If you open your stomata, you're going to get CO2 exchange. You're going to get rid of your oxygen, so your enzyme's not a problem. But you're also going to be letting go of your water, which means you're going to dehydrate. You're going to lose your source of electrons, which means you're not going to be able to do your photosynthesis. So you can see basically how this is sort of like a pick your poison kind of a scenario. And this is true for C3 plants, right? So basically, when you have this, your water is going to be trapped inside. When it starts to get really hot and dry, all that water is going to go through those open stomata and you're going to receive a net water loss, which is bad. You don't want that. So what is your response in order to save water and to keep from dehydrating? You close your stomata. The problem is you lose your CO2 and you get oxygen buildup. And so in this particular case, this is going to create the photorespiration scenario. So this reduces over time the efficiency of photosynthesis and will eventually shut it down because rubisco will progressively more and more and more bind oxygen because oxygen is what's around. So you can see how it's a bit of a problem. So as a C3 plant, typically you have to sort of commit basically to the reality that you're going to be slowing photosynthesis down one way or the other. You might as well save your water because that's important. And then when it cools off in the evening, you can open up your stomata and then you can kind of let your gases exchange and hopefully get some CO2. But this is typically what happens. C3 plants will basically do a couple of things, right? So they basically will fix their carbon using the Calvin cycle, just like we just saw. But typically their stomata will either open or close. That's it. There's no in between. And so as a result, they have to choose between losing water or photorespiration, basically. So because of their Rubisco enzyme. Now, because of this, this basically means that C3 plants tend to grow slowly in hot, arid climates. So that's what that basically means. So as a result, then, we have adaptations to photosynthesis, slightly different alterations in the way or the organization of the way that you do photosynthesis. And this is two different types of plants that use what's referred to as a C4 type of photosynthesis and CAM, what's called CAM. And so this basically, in this case, so instead of using a 3-carbon molecule for photosynthesis, you use a 4-carbon molecule for photosynthesis in a C4 plant. So what happens here is you add a CO2 to phosphoenol pyruvate, and that's going to form a 4-carbon molecule. That's the reason why it's called C4 in both cases. It also uses a totally different enzyme for this fixation. So this is a different, more faithful enzyme to CO2 for carbon fixation. And that's kind of what you're talking about here. You're talking about innovations in carbon fixation. So rather than using, kind of the philandering bum of an enzyme in Rubisco who equally binds to CO2 and oxygen, pup carboxylase is an enzyme that will only bind to CO2. It has absolutely no affinity whatsoever for oxygen. So this is a sort of situation where, like, you take, say, for instance, a woman who maybe have a husband who's very faithful to her and is very much so. So this woman could basically take him into a room full of all these women, and he's like, no, don't even register with me, because I only have eyes for that one woman, the one I'm married to. So pep carboxylase is a very faithful enzyme to CO2. It has no affinity whatsoever, does not care at all about oxygen, only is looking for CO2. And that's important, right? So it only has that one side to it, which is what you want, and no oxidase activity, no that oxygenase stuff. And so both of these C4 and CAM plants will use pep carboxylase as a more faithful enzyme to actually do their carbon fixation, which is critical. And it's a huge innovation over C3 plants. So in C4, then, what you have is a way to spatially separate carbon fixation. So they have a spatial solution to the separation of carbon fixation. And the idea is once you fix it into the system, then you can basically send that through the Calvin cycle. In CAM, you have a temporal solution to the separation of carbon fixation. And so it's a slightly different strategy. So let's take a look at this here. First of all, we can see a C3 pathway. So for instance, columbine, and we should all be familiar with columbines. And so this is a situation where our carbon fixation is fixed into the Calvin cycle with Rubisco directly into the Calvin cycle. So this here is a mesophyll cell. This is one of these, either these guys here or the parenchymal cells, but this is a mesophyll cell. So you can see the carbon fixation is happening inside the cell in C3. And that's your traditional C3 situation. And of course, that basically leaves your stomata having to either open or close. In a C4 pathway, it's a little bit different. Notice you have the C4 leaf is organized a little bit differently. So you can see this little ring of mesophyll cells around your bundle sheath cells. And so the idea here is carbon fixation is happening in the mesophyll cells. So you have an initial fixation in the mesophyll cells using the pep carboxylase, which grabs onto that CO2, any little wisp of CO2, by the way. Even if you have just a very little bit of CO2, that pep carboxylase is going to grab onto that CO2 and it's going to dump it into this 4-carbon molecule, which is the reason why we call it C4. And then this 4-carbon molecule is then transported into the bundle sheath cell where you're going to find the Calvin cycle, and then it's going to be basically dumped into the Calvin cycle. So you can see basically you have almost kind of like a harvesting, almost like a carbon harvesting type of a system with your mesophyll cells snatching onto those CO2 molecules very, very aggressively, very quickly. If you take a look at C4 plants, so we have some common C4 plants, right? Corn is a C4 plant, grows very, very fast in hot, arid climates. Also, weeds are typically usually C4 plants. What are types of C3 plants? Usually flowers are C3 plants, grasses are C3 plants. We can see this because when it starts getting hot, grasses they don't grow very well, right? They're kind of like, they just sort of gas out and their growth rate slows down. The reason is because they're having to close their stomata to preserve water. And so they're not able to do photosynthesis at their normal rate. When you talk about C4 plants, what this basically allows them to do, the combination of PEP carboxylase and the spatial organization of their initial fixation of that CO2 allows them to be able to leave their stomata open just a little bit, right? Because the idea here is usually in the C3 plants, this is your fate. You're either wide open, here's your open stomata, right? Or you are closed. There's really no in between. Now, in a C4, because you have this initial fixation step in the mesophyll cells, what this allows you in the peptide carboxylase, what this allows you to do is it allows you to be able to toggle between your wide open, which is good for everybody, or you're partially open. So because you don't need a lot of CO2 in order to keep yourself going, you can basically close yourself off a little bit, preserve some water, and still get a little trickle of CO2 in there because your PEP carboxylase is so aggressive and has such a high affinity for CO2 that it's going to grab onto whatever CO2 slithers in there, and it's going to be able to still keep your photosynthesis going. This is one of the reasons why we see C3 plants, grasses for instance, stalling out when we hit the July heat. And corn, right, knee high by the 4th of July, is basically getting ready to just take off, is because they're starting to get an advantage now over the C3s because now they're still able to keep doing their photosynthesis when C3s are having to sort of stop production. And so that's kind of our C4 plants, right? So we have corn, sugarcane, sorghum, things like that. So lots of different sort of very weedy growing types of things. The important thing is that initial step is driven by PEP carboxylase. Remember, that makes all the difference. And what it does is it basically takes this carbon and it converts it into oxaloacetate, which then goes into malate, which goes into the bundle sheath cell. So these are both 4-carbon molecules. We already saw oxaloacetate in the Krebs cycle, right? So this is a 4-carbon molecule that gets converted into another Krebs cycle 4-carbon molecule, which is malate. And then that goes into bundle sheath cells, which is where the malate then gets decarboxylated. So you get a CO2 coming off of that, and that CO2 will go into the Calvin cycle where we do have Rubisco. But guess what? We don't have to worry about it because there's nothing but CO2 in there. Rubisco never sees oxygen. So that's kind of what kind of sets this up is with Calvin cycle, we're stuck with Rubisco, but the way we feed Rubisco nothing but CO2 is by creating this initial fixation. So we gather up only CO2 and we're feeding Rubisco only CO2. It never gets to see oxygen. So it's kind of what it looks like graphically. So here you can see your CO2s coming in. Your PEP carboxylase is basically going to take PEP phospholipyruvate. It's going to add that CO2 on there, and you're going to get oxaloacetate. Oxaloacetate then is going to get converted to malate, which then goes into bundle sheath cell, gets rid of its CO2, which enters into the Calvin cycle, gets fixed into the Calvin cycle by Rubisco, and then it turns this into pyruvate. And then this goes back into the mesophyll cell where the pyruvate is phosphorylated by an ATP to generate phospholipyruvate. And so you can go pick up another molecule of CO2. So this way, you never really have the oxygen exposure to Rubisco because you've got this spatial separation. You got the carbon fixation chamber, which is designed to feed nothing but pure carbon to Rubisco, so it doesn't have a choice. but to be faithful to CO2, because CO2 at the end of the day is all Rubisco ever sees because of this organization. So this is where you get rid of or get over that photorespiration piece. However, it is expensive, right? Because you have to generate ATP in order to do it. So you have to have a good, strong, robust energy plan in order to pull off a C4, which is one of the reasons why C4 plants are energy hogs. I mean, if you basically take a look at your C4 plants, your weeds growing in the yard, you'll notice that they're basically sucking up all the nutrients and they're sucking up all the energy in the soil, leaving your poor little C3 plants sort of gasping for air because they're getting all the nutrients taken from them. So that's part of the energetic cost associated with driving the C4 pathway. And so this is perfect, right? So when you have a hot, dry climate, you're able to still maintain your open stomata because you have a faithful carbon fixer, PEP carboxylase. This allows you to be able to continue doing photosynthesis even though it's a hot, dry climate. Whereas a C3 pathway has no choice but to shut down the stomata. That's going to shut down photosynthesis, which means they're not going to be able to grow nearly as fast in the hot, dry climates as, say, for instance, a C4 plant. Okay, what about cam plants? Well, cam plants have kind of a different thing because remember these guys are going to be temporal, night day time, right? Temporal separation. That's what they're going to do. And so these basically are the succulents. So if you have kind of a fat, fleshy leaf like aloe plants or cactus, things like that, pineapples, for instance, are a type of cam plant. These are all cam plants. So here's what they do. So instead of separating carbon fixation spatially, what they do is they do it temporally. So during the night, the stomata will be open when it's cool and there's low water loss. So basically, less water loss. So they can open up their stomata at night and not have a lot of consequence to losing water. Then when it starts getting daytime, they're going to close their stomata to basically prevent water loss, which is the opposite of what most plants do. Most plants will open it during the day, close it during the night. So what does this do? What this basically does then is during the night, actually, yes, during the night, open stomata take in and fix CO2 without water loss. And so they're going to fix their CO2 using, again, PEP carboxylase, and they're going to store that that molecule, right? That 4-carbon molecule, they're going to store the product in one of their vacuoles. So they're basically taking in as much CO2 as possible in the night without their water loss, and they're basically storing it all up. And then what they do is they take the storage, they close their stomata at night. So closed stomata prevents water loss. And then it allows CO2 fixation, right, from stored CO2 during the day. So what they're doing is they're basically soaking up all their CO2 at night, and then they're just going through their fixation, their CO2 fixation during the day. And then they're able to generate there. So they're basically circumstantially or they're temporarily opening up their stomata. And so that's what's happening. So when you're closed during the day, you're making your sugars. When you're open at night, you're collecting in your CO2. And then that's different than the light reactions, right? Because the light reactions are going whenever there's light out. You're just basically making and storing ATP and NADPH. And then whenever you're doing the Calvin cycle and the stroma during the day, then you're able to just basically consume that. So the light reactions aren't really affected. This is basically associated with the Calvin cycle for the most part. And so you basically have these high levels of stored CO2 that have been converted or fixed in this system. And then you basically use that in the Calvin cycle throughout the day. And as a result, then you eliminate that photorespiration. You can have your stomata closed during the day, and then save your water, and then you can still do everything you need to do. So oftentimes then, when you take a look at cam plants, you have some, reminiscences of C3, but you also have C4 as well because you're using PEP carboxylase. So in C4 you have two pathways: fixation and then the Calvin cycle happening in different cells. And in CAM you have basically the C4 pathway at night, which is the carbon fixation, right? And then the C3 pathway, which is dumping that molecule into the Calvin cycle, happening during the day when your stomata are closed. And so that is basically chapter 8.